Hello and welcome. I'll begin by talking about the features that discriminate the MIT-1000 RMID system from other microbial identification methods. You will see most of these features discussed and demonstrated in this presentation. The MIT-1000 technology does not rely on biological cues. Consequently, chemicals, reagents, or species-specific dyes are not used at any time. The system identifies by measuring laser light scattered off individual microbial cells. The laser is the same as used in standard grid laser pointers. However, the laser intensity is 10 to 15 times brighter. The system uses digital libraries called identifiers during the identification test. The identifiers are lookup values used by a statistical classification algorithm developed by us at MIT. Each individual identifier contains information specific to a microbial genus, species, or strain. New identifiers are easily created and added to the MIT-1000. Sample preparation is particularly simple, consisting of 6 easy steps with a preparation time of about 1 minute. Inexperienced laboratory technicians have learned the procedure in about 1 hour. Sample preparation costs are very low. The few consumables are items that nearly every microbiology laboratory uses every day during its normal operation. Once the sample is in the system, beginning the test is a one-button click on the graphical user interface. Identification test time is very fast. Typical test times are less than 5 minutes, and some tests are as fast as 30 seconds. The system provides unambiguous results. Secondary refining tests are not required. I'll say much more on each of these features in the following slides. The entire identification test procedure is very simple. First, make a sample. The sample is a very weak dilution of individual cells taken from a colony on a culture plate, suspended in filtered water in a round-bottom flask. Each cell in the source colony are all genetically the same. Next, place the sample vial in the MIT-1000 system. Then, select the microbial types to test for by selecting the appropriate identifiers. The selected identifiers are called active identifiers. The system will only identify the microbes handled or managed by the active identifiers. The active identifiers from the previous test are already selected, so typically this test has already been achieved. Any number of identifiers can be active simultaneously. Click Identify on the MIT-1000 application's graphical user interface. After the user enters some sample-specific information, the test begins. The system examines and evaluates each detected and measured cell as it passes through the laser beam. The measured data for each cell is combined with the lookup values in the active identifiers. The typical test time is less than 5 minutes. The system measures as few as 10 to as many as 100 cells before it draws its final conclusion. The system then stops the test and displays the final result and is ready for the next sample. This is what the optical platform of the MIT-1000 system looks like. It consists of 5 concentric arcs with as many as 8 single-element light detectors mounted on each arc for a total of 35 light detectors. Here, 3 arcs can plainly be seen, here and here underneath the green, and here. The green rectangles are circuit boards for the individual light detector chips. And you can see here the top of the sample vial, and in the back is the laser. The signal measured by the light detectors pass along the gray cables to the digital interface board mounted on the back of the system, just visible here. The system connects to a Windows PC through a USB interface. Here's a bare-bones schematic of the optical platform. This shows only the arcs in the sample vial. 4 of the arcs are positioned like an X when viewed along the optical axis of the laser beam. The 5th arc is at the bottom. The size of the arc structure is about the size of a soccer ball. Clearly visible here are the light detector mounting ports. The detectors view down through the small diameter hole in the arc to the center of curvature of the arc, so each light detector views the same point at the center. The sample vial is a round bottom flask. The round bottom is a sphere, and the vial is positioned so that the center of the sphere is also at the center of curvature of the arcs. And then finally, the laser beam is oriented so that it also passes through the same center of curvature. The common field of view of the detectors is a volume of approximately 1/10 of an inch in diameter. When a bacterial cell passes through the laser beam, the detectors will see it only if it occurs within the 1/10 inch diameter common region. When the cell does pass through the laser beam in the common region, the cell scatters light radially throughout the surrounding space. The light does not simply reflect off the cell. The light penetrates the cell, interacts with all of its parts and features, sending light in all directions in a way that is characteristic to its genus, species, and strain. Some of the scattered light is captured by and measured by the light detectors, digitized, and then passed on to the computer. Going into the details of the identifiers is beyond the scope of this presentation, and beyond that, as you can imagine, the details are very proprietary. Identifiers are the essential product of MIT. They give the MIT-1000 the ability to discriminate one microbe type from another. Identifiers are made by us at MIT. They are built out of an analysis of light scattering data in a large database of characterization measurements we have made and continue to make. These measurements are just like identification tests, except the signal measurements are archived in a database. Each identifier is constructed from an analysis of many thousands of measurements of a large variety of different microbial genus, species, and strains. The analysis looks for those scattering characteristics that can be used to distinguish between a given target microbe type and all other types. The result is an identifier for the target microbe type. The MIT-1000 system uses the identifier to answer the question, does the sample being tested contain the microbial type X? For example, the E. coli O157H7 identifier answers the question, is the sample being tested a member of the bacterial strain E. coli O157H7? The answer is either yes or no. The system allows the use of any number of identifiers simultaneously. So for example, if the user selects the E. coli O157H7, the Listeria genus, and Salmonella genus identifiers and performs an identification test, the test will answer the question, is the colony being tested E. coli O157H7, Listeria genus, or Salmonella genus? The answer is, it is E. coli O157H7, or it is Listeria genus, or it is Salmonella genus, or it is neither, in which case it would give a result of unknown. Now let's talk about making a sample for an identification test. Making the sample uses 5 consumable items: a microcentrifuge tube, a 1 microliter inoculating loop, a pipette tip, a small square of parafilm, and filtered water. Parafilm is a wax-like film used like friction tape. We use it to seal the sample vial. We also use disposable inoculating loops in our lab. A reusable loop could be used. Making the sample uses 4 reusable items: a vortex mixer, gloves, a 1/2 to 10 microliter variable pipette, and an MIT-1000 RMI-80 sample vial. The overall majority of bacterial identification methods begin with colonies on culture plates. Our method also begins there. Here's a photograph of a culture plate with bacteria growing on it. These individual gray spots are colonies of bacteria. A colony is a visible cluster of cells, all descended from a single cell. Since the cells descend from a single cell, they are genetically identical. The colonies are a result of the procedure used to streak the sterile culture plate with a microbial specimen. The first step is to lightly scrape a single colony off the culture plate with the inoculating loop. Step 2 is to dislodge the colony on the loop into a microcentrifuge tube filled with about 0.2 milliliters of filtered water. We use a half milliliter disposable tube with a snap seal. We insert the loop into the water and rub and stir the loop until the colony comes off. Then the cover is snap closed. In step 3, the microcentrifuge tube is vigorously agitated with a vortex mixer for about 20 seconds. This breaks up the colony mass into individual cells. This is especially important for species that like to stick together in chains or in clumps of cells. Step 4, pipette 1 microliter of the dilution into a sample vial that is filled with filtered water. Next, cover the sample vial with a square parafilm, and then finally put the inoculated sample vial into the MIT003A test chamber. Our sample preparation procedure makes sample vials with only a few thousand bacterial cells per cubic centimeter. And at this concentration, a cell will pass through the laser beam once every few seconds on average. The actual rate depends on the concentration. The sample is in position and ready for an identification test.